New York Bio brings you the promise, potential, and policy implications of cell and gene therapies from our 2021 annual meeting brought to you by Vertex Pharmaceuticals, featuring Ginger Davis, the president of the Sickle Cell Thalassemia Patients Network, Assemblywoman Alicia Heineman from the state of New York, and Scott McGowan from Vertex Pharmaceuticals.
That this has been, I think, a very recent period of advancement in sickle cell disease therapies. For the more than 100 years since we did identify what the underlying kind of biological mechanism of sickle cell disease was, there had been really minimal innovation, and you had a drug called hydroxyurea, first approved for sickle cell disease and starting use in the 1980s, and then about a 30-plus year gulf where there were no new therapies for patients living with sickle cell disease. That changed with the approval of L-glutamine in 2018, and then two agents in what was affectionately known as the November to Remember in November of 2019, which again brought new therapies, new options, and new hope for patients. At the same time, there are now dozens of programs in sickle cell disease that are currently in the clinic that companies like ours and others are bringing forward, and it is such a remarkable thing to think about the potential that this pipeline holds for patients, their families, and others. And because at the end of the day, this is a community that, like all rare disease communities, deserves options, deserves choices, and deserves the potential to live better lives.
Pipeline, and it's taken over 100 years to get us to this point, mainly because of the perception that sickle cell disease is a Black disease. In our organization, the Sickle Cell Thalassemia Patients Network, we say sickle cell disease is a human disease. We don't leave out anybody. We don't leave out the Mediterranean, the Middle East, the Southern Chinese, Southeast Asians, the Indians, Central and South Americans who were also affected, along with people of African descent. I am a first-generation American of Caribbean Latino parents. I am the second child born into the family with sickle cell disease, but the one that survived. My brother did not benefit from newborn screening and being identified and died of a treatable complication. So I was diagnosed at the age of 2 after my second crisis. And from there, I was put into comprehensive treatment along with children who had cystic fibrosis, Cooley's anemia, hemophilia, even asthmatics. All of us were thrown in together, and we were a family. I decided at 10, after that conversation, that I was not going to let this disease impact me negatively. I was not going to spend the rest of my life in and out of the hospital. And it's the same with all rare diseases. There's a lot of focus now being put on the disease, but if people do not know that they're carriers, if they're not being tested and not being identified, not receiving genetic counseling for them to understand, and if the information about traits are not up to date, it's a problem.
So I'm a New York State Assemblywoman Alicia Heineman. I represent a large Southeast Queens community, and I represent a large African American population. I am the parent of a daughter with a sickle cell trait. And coming to the assembly, it was important for me to be a voice for sickle cell research, sickle cell funding, and the eradication of the disease. I know we lag behind other states when it comes to funding of sickle cell disease compared to the African American population that we have, but I have to say it is thanks to the advocacy of the groups in New York State for the time that you sat in the lengthy health hearings, the work that you've done outreach to members throughout the state of New York on the Senate and the Assembly, that we are even at this point.
Potential to live better lives. What I'm going to be talking about a bit is the field of genetic therapies, and I feel very fortunate to be able to work with such an amazing community and also to be an ambassador for this type of science, because it is quite simply one of the most amazing potential technologies that we can apply to address diseases like sickle cell disease, like beta thalassemia, and like so many others that currently lack adequate treatment options. In order to start this journey down cell and gene therapy, it's important to take a step back and realize that all of this is building upon decades of scientific advancement. We have, obviously, with the sequencing of the human genome, with advancement in science, we've been able to not just understand what's happening in disease, but where it starts. And for genetic diseases, that begins at the gene. And our quick survey of folks in the room, who knows what a gene is? Okay, so we've got a fairly wide representation. I will still give you a moment of high school biology, which is that genes are often called the blueprints for life. They encode something called proteins, which is a type of molecule that you need for your cells to function normally. And so when you have a mutation or an alternate form of a gene that is not the standard, that can cause disease in some cases. And in the case of sickle cell disease, it is a single mutation in a molecule that encodes part of hemoglobin, which is what carries oxygen in your red blood cells, drops it off of the tissues, and makes sure that your body essentially can breathe all throughout the body. Genetic therapies will target then, therefore, that gene. When you want to go to the root cause, you need to go to that gene. Otherwise, you're treating upstream, you're potentially treating symptoms, and it may not be able to arrest, solve, or otherwise correct what's happening there. So that's where cell and gene therapies really come in. This is not necessarily a new field, as we've started to learn more and more about genes, genetic disease, and the genetic roots of really so many things about the human body. We've been studying this in the clinic since about 1989. That was the first ever gene replacement therapy trial that occurred in a disease called SCID, which is otherwise known as kind of bubble boy syndrome, where they have no functioning immune system, have to be completely blocked off from the world. What Vertex is doing specifically is a type of technology called gene editing. And if we keep this metaphor in our minds of genes as the blueprints for life, genes as the instruction manual for how you build your body, if that's the case, then gene editing is kind of like, if you imagine a Word document for that instruction book, being able to use the find and replace function. So you may have a genetic code that says, I need to go let in the cat. Now, if you have a single typo in that, a single error that says, I need to go let in the bat, well, that's a very different situation. You probably don't want to bring a bat into your home, but again, you can imagine how that could easily be done. So if you were able to use gene editing, a specific molecular tool, to go in and make that one change to the reading, then it goes and returns back to, I need to go let in the cat. That is the type of precision that these approaches offer. It is something that we have been working on in the laboratories for the last decade. And one of the reasons you saw this technology be awarded the Nobel Prize in chemistry a year and a half ago to Drs. Doudna and Charpentier. So it really is an incredibly powerful tool, something that has applications not only in human health, but also in agriculture, industry, and so many other places. So what we are endeavoring to do is to essentially change the way the body makes its own red blood cells by providing them with new bone marrow that is taken from theirs, edited, and then provided back to the patient. These are the types of approaches that are currently being studied in the clinic. Now, I think acknowledging that we have a great group of stakeholders here who represent not only policymakers, sponsors, partners, patients, and others, is that our healthcare system is not really set up right now to accommodate these types of potentially curative genetic therapies. We are used to a construct in which we are chronically dosing medicines, be they biologics, be they drugs, in a manner that we pay for in kind of units of service. This is more akin to something like a genetic surgery, where we're going in and potentially fixing the root cause of disease to provide hopefully lifelong benefit. And those distinguishing characteristics make this type of approach very different. So I think it is really incumbent upon all of us to really recognize that this is not something that is potentially decades in the future. This is something that we are thankfully on the cusp of a medical revolution in this type of technology, and we need to work together to ensure that not only patients on private insurers can benefit from these, but really all patients. It's I think, reasonable to recognize here that cell and gene therapies are going to be very expensive therapies. We have conversations, and rightfully so, about high cost of drugs in this country and trying to figure out how are we providing therapeutic options that are really options for patients, that they can benefit from, and that they can be paid for in a way that recognizes the value they present, the value of innovation, and allows us to continue this biomedical research engine to churn out new therapies and new cures. We need to think differently, think big, and be as innovative in our commercial strategies as we are in our labs. And so some of the things have been outcomes-based arrangements or value-based arrangements, whereby a payer, be it a Medicaid, be it a private insurer, are going to only pay if certain metrics are met, if it is shown to be effective and durable over a set number of years. These are some of the things that folks have looked at. Also, reinsurance programs, whereby you're able to share risk across a broader risk pool for these, probably lower probability events, these less frequent events where you'd have a cell and gene therapy eligible patient, but still something that enables people to have access. Because at the end of the day, if we have patients who are unable to access these types of therapies but could benefit from them, then we as a stakeholder community and we as a society have not succeeded in our mission.
That's happening right here, right now, in this country. We're talk about what's happening in the other parts of the world, but if you have a parent who's working and has insurance through their job, one of the approved medications in 2019 comes in at $127,000 annually. The copay for that, for some of them, are $9,000. Families can't manage those payments, So, and other things, when doctors go in to talk about the medication to payers, there'll be a sixth statin, does the same thing as all the rest of them, fine, it passed. You talk about sickle cell disease, and here's a new medication. It gets slapped with a pre-authorization. Mm-hmm. That is a barrier to accessibility. So majority of our population is on Medicaid, Medicaid managed care, and Medicare. And sickle cell is the most expensive condition to treat, more so than HIV/AIDS and cancer in this country. And we can show enough statistics where if people are getting preventative care and we can keep them out of the ER and out of the hospital, the money that we're asking for treatment centers throughout the state, it would be made back in the first year.
And I just want to thank NewYorkBIO for having this forum and for keeping this at the forefront when it comes to gene and cell therapies for sickle cell patients. That it's very important that we include and not be exclusive in the community work that we do across the lines to make sure that we move the needle, like I said, forward to what we need to do for eradicating sickle cell disease.
Thank you for joining us for the promise, potential, and policy implications of cell and gene therapies. Thank you to our panelists and to our colleagues at Vertex for sponsoring this event. For more from our annual meeting, please follow us on Twitter at NewYorkBIO and on LinkedIn.